Hi everybody, I'm really glad you could share your Wednesday afternoon with me. I am delighted to be here. I've been working as part of UCT now for a couple of years actually. I spend my Tuesdays over at the med school campus working with Jonathan Blackburn's team. So part of the funding that brought me here is from the Medical Research Council, and their goal is to see that everybody gets a little more bioinformatics in their lives. So I'm glad to work with you. If you're running into some problems and figuring out how to wrest information out of your data sets, give me a call. We'll see what we can do. My last name is Tab. It's a pretty distinctive one, so I don't think there are a lot of us in South Africa yet, so feel free to write. So I was asked, what am I doing as a basis for how to frame my talk today? And I thought that it would be best to talk about a set of projects that all fall on this boundary between genomics and proteomics. I spent a lot of time thinking about just proteomic identification. That's really the field that brought my career to a boil, creating the algorithms that let us identify proteins and identify peptides and post-translational modifications from tandem mass spectrometry data. And that's a place I'd love to talk for a long, long time about. But today, we're going to change gears a little bit and talk about this touching the world of genomics. How do we draw information from genomics into proteomics? And what can we do in proteomics without genomics having been done? So it should be a lot of fun. I'm going to start with kind of a classical application. Now it's kind of nonsensical to talk about the term classical in the same sentence with proteogenomics, but this is what most people think of when they start talking about proteogenomics, which is to say, find out whether or not genomic variants are expressed, figure out if there are ways that we can improve our gene definitions within genomic data. So I'll be talking about that primarily in the context of TB. Hope you'll forgive me, that's really what the SATB program is all about. But from there, we're going to launch off into a rather different direction, and that is the question of orthology alignment. This is kind of a new approach that we've been trying to pioneer, largely in connection with the University of the Western Cape, because I gotta say, they're in a world of hurt because they're working in plants, and plants are a much less tractable place for genomics in a lot of respects. So we'll talk about that problem in a moment. And finally, we'll move on to pre-genome proteomics. There's this claim from genomics people that proteomics people are always the followers. You know, you go into a new species with your genomics tools, and once the ground is safe and you've mapped out all of this uncertain land, then and only then can you do proteomics. Well, we're out to change some of that, so watch out. So on we go. Proteogenomics. So what are we doing with this in the space of TB? Well, as I think a lot of you will know, tuberculosis has all kinds of interesting strains that are evolving in human populations, and a lot of them are right here in South Africa. Now, a couple of the strain variants that we were working with at Stellenbosch University are called LAM, or Latin American or Mediterranean family. So we had a couple isolates of this LAM type that we were looking at from a genomic point of view, and we wanted to see if any of these differences that we could detect at the genomic level were also making themselves felt at a proteomic level. We had three different starting strains that I tend to call these saucy, so I hope you'll pardon me. Saucy 3651, Saucy 3517, and H37RV, which is our common laboratory reference strain. Each of those we produced 2 biological replicates from, and then for each of those biological replicates, we had produced what's called a gel-CMS experiment, a gel-CMS. So this is a case where you separate the proteins out on a 1D gel, and then chop up major sections of that into tiny little diced pieces, digest the peptides out of that, and run, in this case, 10 different LC-MS-MS experiments from each gel. So if you look at this, you see that there are 12 gel-CMS experiments, each of those spanning 10 LC-MS-MS files. So this spans 120 raw files. It's one of the larger data sets that I've encountered here because very frequently in South Africa, I see that people do not use fractionation in conjunction with their proteomics experiments because it's expensive. Suddenly, instead of paying around $100 for an LC-MS-MS experiment, you're paying $1,000 for 10 shots. So that is an expensive proposition. On the other hand, this approach, this of gel-CMS, lets us look much deeper into these proteomes and get much better sensitivity. Okay, so we had these data. We knew that they had major deletions that separated these 2 strains from H37RV, and they even had some pretty significant variations between the two as well. We had the data collected on our Orbitrap Velos over at Stellenbosch University, and it was time to put all those data to the test. Now, happily, this work's been published, so I don't really have to worry about telling you any part of the story that hasn't been told already. So let's start by saying proteogenomics is kind of a complex place to be from a bioinformatic perspective. If you look at this diagram, you can see that we had our pile of 120 raw files from an Orbitrap Velos on the left, and on the right, we had whole genome sequences that were produced by our collaborator up in Saudi Arabia. So on the proteomic side, we need to do a bit of quality control. We need to figure out if any of these files it represents a failed experiment. I mean, we might need to rerun one of the gel CMS experiments. We need to convert them to a format that lets us process those data a little more flexibly. We need to do database search. This is kind of the time, the costly time step of proteomics, the ID step, figuring out which peptides each spectrum best represents. And finally, building a protein inference list that covers all of these data and helps us to see where are the evidence for this peptide or this protein across all 120 files. So it's a fairly complex inference and data management step. Over on the genomic side, every project should start with quality control, and that's true for sequencing as well. We used FastQC to evaluate the sequence read quality. Pretty good stuff. I've left out a step here, trimming off the bits of sequence that are not actually helpful. We had read aligners, so we needed to be able to see where within the MTB genome these reads fit. And that gave us sections of sequence that were aligned successfully and may have contained variants which needed to be called out by different algorithms. And we had other bits of DNA read that didn't align to this reference genome. And that's kind of a problem of its own, isn't it? So here we've got this set of sequences that do not seemingly correspond to MTB. This stuff happens. So for that, we use something called a de novo assembler to try to create a contig that spanned all these reads that we had produced that didn't align to the reference genome. So that's a lot of work. But at the end of the day, we were able to flow 3 different kinds of sequence databases across. One would be the reference proteome. We already know all of the proteins that should be found in H37RV. That's kind of our base for all additions later on. The next would be adding variant peptides. This is a case where we had sequencing reads that mapped to the genome of MTB, but they showed that there were some non-synonymous single nucleotide variations. So we were able to see places where, for example, a leucine in the standard H37RV had become a valine in these 2 lamb strains. But each of the lamb strains had their own peculiarities. So we were able to create a sequence variant FASTA sequence database as well. And finally, we were able to stick on a whole bunch of added sequences that were found in these unmapped regions. Because these unmapped regions may contain all kinds of stuff that ends up getting expressed. And if we could confirm that any of that were expressed, we would feel really good about it. So there's a whole lot of data flow here, but all told, they allowed us to tell a much more interesting story about the data we had collected. So how much impact does this have? We started with our basic set of sequences for H37RV, 4,000 plus. Here we have just a few more that we've added that contain variant peptide sequences where we saw that the genome was arguing for a different letter at these positions than we would have expected. We also were able to add in 6-frame translations of these large unmapped regions that we disassembled from the reads. So we had some sections that looked like they had pretty good open reading frames. Can we then do a 6-frame translation to get out the amino acid equivalent of those sequences? Because maybe we'll get a hit, you never know. As you can see, these have an impact on database size. Here we've risen from about 4,000 to, sorry, I think it's about not worse than 2,000 additional sequences for that. But what happens if you were just to start with the reference H37RV and do a 6-frame translation of all of that? Well, things go off the rails really, really quickly. As you can see, the number of protein sequences explodes to the right if you attempt to do a 6-frame of everything. And for a lot of people who are doing their first proteogenomics experiment, they attempt this 6-frame translation approach and realize that they're in over their heads very, very quickly. So we needed to limit the amount that our search space grew by doing proteogenomics. All right. Now, I said that we collected these on our Orbitrap Velos, and the tandem mass spectra, the evidence for individual peptides, was collected in this, via CID, so low resolution, low mass accuracy fragment ions. And a lot of people would say, well, now hold on, how are you doing anything with proteogenomics if you're going to be dealing with this large search space? Must you not have high resolution tandem mass spectra? Well, we still had accurate precursor masses, we just had low resolution fragment ions. And I would ask you, do the identifications we produced look implausible? I think you would argue that they don't. We were able to find dozens of peptides that showed that these sequence variants don't just exist in the genome, but these are actually expressed. These become real proteins. And that was very encouraging. But because so many people have published so much nonsense in the space of proteogenomics, this reference down at the bottom from Alexei Nesvishny is a very good reference to look at to see why. We decided that we would manually inspect each one. We would manually inspect each of the tandem mass spectra for these variant peptides. And we felt that 59 of them passed. They were very convincing spectra. That's not to say that the others were trash, but the others were not as convincing as these. So at base, we feel that we have very compelling evidence that we can recognize expressed genetic variation in MTB strains at the sequence level. So we felt very encouraged by that result. And so did our peer reviewers, which is always a good thing. So finally, what about all that unmapped six-frame translation stuff? We were surprised to discover that we got lots of hits in this unmapped region. In fact, we had a particular region of the sequence that produced 29 peptides. That is not likely to happen by random chance. It's a vanishingly small percentage. When we look at the spectra, we see that they're quite beautiful. The intensity that we see in the tandem mass spectra aligns very well with the fragments that have been matched from this peptide sequence. What we discovered is that these hits all come from the region of what appears to be a novel helicase that was part of one of the lamb strains but not part of the other. And it's certainly not found in H37RV. So we can see that these strain variants don't just change around the sequences slightly of the genes that they're carrying. They may occasionally perhaps have horizontal gene transfer. They can gain protein sequences that we don't associate with H37RV at all. So that was kind of a surprise for us. All right. So that's kind of a classical exploration of the kinds of things one expects to do with proteogenomics. To find genomic variation is confirmed to be gene expressed, and to have the evidence on a sequence level that shows that this particular sequence is available. When we switch gears into the fine world of agriculture, we sometimes find that we're in a very difficult place. A lot of the proteomes that we work with routinely, like H37RV, like human, like mice for that matter, or yeast, that these things have been tread over so many times that they've got excellent annotations, and there's not a whole lot that one can second guess about them. But as soon as you move into the world of plants, you'll frequently find that the genome annotations are hugely problematic to produce in the first place, and the sequence identification from them can be quite a lot more difficult. So the problem I'm talking about today is one of doing proteomics, actually differential proteomics, in two species at the same time. So imagine if you are studying a particular cellular system in humans and you have a mouse model. You might like to see that the mouse and human proteins respond in a similar way between these two. It's not that different to be doing it in the world of agriculture. So here we have two C4 photosystem plants. We have maize and we have sorghum. We're working with sequence databases from Phytosome, which is a big database made available to us from the Department of Energy over in the States. The University of Western Cape was interested in evaluating the performance of the proteins from these two organisms in well-watered conditions and in drought-like conditions. Because as it turns out, sorghum is pretty good at dealing with water crises, but maize, not so much. So, wouldn't it be nice to either be able to find out how we could tweak maize so it was better able to resist drought, or to find ways to make sorghum more productive, for example, to make it a more economically viable crop. So, we had 5 replicates in each of these boxes. So 5 replicates of well-watered maize, 5 replicates of drought-like condition for maize, and the same for sorghum. But here's the problem. We want to be able to see that the same gene in maize is behaving like its relative in sorghum. So we call these orthologs. The same gene in different species, those are orthologs. So we needed to have a map, an orthology map, that let us say that for this protein in sorghum, this protein in maize is its best ortholog. It sounds simple, but in fact it's kind of a hard thing to get at. Oh, so these data were all produced over at CPGR. I've just included their logo without the name, but CPGR generated the 20 raw files corresponding to this experiment. So we decided on a 2-step process for this. The first would be establishing this map of orthology between the whole proteome sets of these 2 species. So how big are these? So if you're working with human, you might have upwards of 70,000 protein sequences. If you're dealing with MTB, 400 or so, right? So dealing with plants, you generally get into pretty big search spaces. So with sorghum, we saw that the reference database at present contains 47,000 or so sequences, protein sequences. One of the things that this suggests to me is that we haven't done a lot to study all the isoforms that can be found for a given gene in this species. We've done a little better, I think, in the space of maize, where we have 89,000 protein sequences to work from. So how do we go about finding orthologs? I think everyone here has probably encountered BLAST at some point in their lives. Right, so BLAST is really good, but you don't have to use the web server. You can, in fact, download BLAST and run it on databases there on your own computer. And we decided we needed to do that because in this case, we were going to search for every sorghum protein in maize. And we were going to search for every maize protein in sorghum. What we were looking for is something called a reciprocal best hit. Reciprocal best hit. This is a fairly conservative strategy, but one that helps to resist erroneously claiming that 2 proteins are actually relatives of each other. So if we were going to call a protein a best hit, we needed protein A in sorghum to hit best to this particular sequence B in maize, and when we did the corresponding search, that protein B in maize would specify A in sorghum as its best hit. So you end up with kind of this awkward arrangement. Now imagine that these run on for 89,000 entries, right? We have our sorghum proteins up here, we have our maize proteins down below. You may have a brilliant situation in which the maize protein points to the sorghum protein points to the maize protein. Brilliant. For about a third of the sorghum hits, as I recall, we had this kind of relationship. So that was very reassuring. But you can end up with some uncomfortable situations. How about this one? Here we have a sorghum hit that has 2 different maize proteins claiming it's the best hit. But the best hit from that maize protein is neither of those. So that is a nasty little problem. So we needed to be, and certainly you can run into proteins that have no analog in the other species. So we needed to be able to deal with that kind of ambiguity. Once we have our orthology mapping table, we can now say this protein should be aligned with that other protein across the 2 species. We can begin working with our protein count data. Now this is a bit messy. I want you to imagine that we have a long table, say 1,000, 2,000 proteins, whatever it is tall, and then we have 20 columns for it. For each protein, we say how many spectra in this particular raw file match to this protein. That's a spectral count table. So we have 20 columns, we have 2,000 rows, whatever the number was. The problem here is that the software sees the maize protein and the sorghum protein as differentiable from each other. They have peptides that allow us to tell one from the other, which means that the sorghum protein and maize protein are going to be listed separately in this massive table. And there's no guarantee they're anywhere close to each other in the ordering. So once we find that both members of an orthology pair are in this table, we can grab these numbers A through J from the sorghum table for the sorghum ortholog and grab the corresponding counts for the maize protein and build a new row that brings them together. And now, when we create a statistical model, it can recognize that both maize and sorghum are available for this pairing. Okay, so that was a little bit of our magic, and it took a little bit of scripting and thought from me. But in the end, we got something that more or less worked, and we're really excited about it. I'm hoping to get a student on this project to look for all the places where I made mistakes, because my coding skills are rusty, let's just say. All right, so why bother with this? Why bother with this? Annotation in plant proteomes is pretty weak at this point. It may be that maize, with a more mature set of data behind it, is a little better annotated at this point. So if you are looking at a particular sorghum protein in the database, you may get no description whatsoever for what this thing is. If you can find, however, that it has an ortholog partner over in maize, and that ortholog partner has a description, you can just transfer that description over and you're happy. Now you have some information that you didn't have before through transfer of annotation. Okay, so that's a very helpful first place to start. Next, statistical models. If you double the amount of information available to your statistical model, it's going to find it a lot easier to reliably find differences in expression between wet and dry. If you have a protein that transitions from low in wet to high in dry conditions in both species, that signal is going to be amplified by having these data unified. Having more information makes statistical decision-making more straightforward. In the end, we found that we were able to double the number of proteins that had aligned maize and sorghum information in our tables. So that made it much more possible for us to ask this question about joint movement between these two species for drought and wet conditions. So I was really happy to see that. Ultimately speaking, we wanted to see how the gene in both species responded to these wet and dry conditions, and now we can do that. Okay, now we're going to move to a more speculative realm. For years, we've had to trail behind those awful people in genomics. I bet there are some here. All right, so we've had to trail behind them, had to pick up their leavings, deal with their half-baked annotations and use them and just say it's got to be good enough, it's what we've got. But it's becoming possible to do good molecular biology in the fine world of non-model organisms. And in this case, our friends over at University of the Western Cape, again, are looking at superfoods. Has anyone eaten chia? Chia seeds, anyone? A few people? Now see, in the United States, we used to see commercials for chia pets. Has anyone heard of these? Chia pets? You would buy a little clay statue, and it has these grooves on it, and you would paint this kind of green paste over the grooves, and then you'd water it every couple days. And sure enough, after a while, you get little sprouts of chia growing out of it. So you could get like a little clay head and like paint green hair on it, and it would have this flowing green mane after a while. So it had a really delightful little marketing jangle. It went something like, cha-cha-cha-chia. Anyway, that's fine. So I always have that song in my head whenever I'm working in this field. That's fine. So we want to do proteomics in chia. UWC is doing proteomics experiments today in chia. But how do we make sense of the data? There's no genome annotated for this critter yet. So that's a bit of a problem. So we did a little bit of digging through the literature. Somebody out there had spent a little bit of time working on the molecular biology of chia. A group in India published a set of 5 transcriptome sets in RNA-seq for this species. And they produced an annotation of all those data and kept it for themselves so that they could write more and more papers about chia with this resource that nobody else had access to. I don't like that attitude, but they made their RNA-seq data available to the public, which meant that we could do the assembly ourselves. So we did. We used some software called Trinity. Trinity is a really widely used toolkit. It is certainly not the only one. There are always people developing new algorithms for particular applications in genomics. So we were able to use Trinity in a kind of an advanced form for it. I think it was version 2.1 or 2.2, something like that. Now the authors of this paper had originally used Trinity on their data sets to produce a list of transcripts. But they produced 76,000 transcripts using exactly the same data and exactly the same, and using a version of the same software. We produced 55,000. So what changed? Maybe they did some trimming differently. Maybe they parameterized the software differently. Maybe they were using an altogether different version, I would assume so. Actually, they didn't tell us what version of the software they were using, so we couldn't replicate their experiment. This is a real problem anytime bioinformatics or biostatistics are involved. So we needed to be able to figure out how these reads could be assimilated into transcripts, and how could we recognize that different isoforms, different transcripts were produced from the same gene, even. Trinity handled all of that for us, so we really appreciate that. That gave us these 55,000 transcripts, but were they any good? How would we know? So the good news is that the fine folks over at CPGR were able to produce some LCM-SMS experiments for us from Chia. And we can use those data to figure out whether or not we're hitting real proteins with these sequences. But just to begin with, we wanted to add some annotation information, to add some description power. So we thought we would use two different routes. One is tBLASTn. tBLASTn lets us start from a transcript and search against a protein. So if we're able to assemble a set of proteins, we can do a search. Now, we started by trying to run tBLASTn on the servers over at NCBI. Now, one of the things a lot of people don't understand about the network is that it's not just that anywhere on Earth is connected to anywhere on Earth. Africa has good bandwidth to Europe, but it does not have good bandwidth to the United States, for example. So every time we sent a tBLASTn search, it was passing to Europe and then across the Atlantic to the United States. This slowed us down quite a lot, actually. And after having a server try for a week to do these tBLASTn searches, the postdoc gave up saying, this is a fool's errand, we don't need to do it this way. So how can we seed a database together for use locally that lets us do BLAST on our desktops to forego this network problem? We decided to go exploring a little bit. We did an examination of different neighboring species that were close to where Chia fits in the hierarchy. So you might think, well, gosh, if you're dealing with a plant, you're going to have something with decent annotation. Maybe you should just throw some Arabidopsis at it, right? It turns out Arabidopsis is pretty far off. It's pretty far away. But we do have 4 completely sequenced species that are pretty close by that we were able to find through the RefSeq resource at NCBI. We start with Paraboia paniculata. Sounds like something you should wave your wand, you know, paniculata. But there we go, an African violet. Next, we have the Genlizia aurea, which I think looks, you know, kind of, I like looking at this because it's been so dry. But if you look at its description, it's a carnivorous plant. For me, that gives it bonus points. Of course we're going to include that in the companion set. Then we get down to Sesame indicum. I have some on my hamburger buns, so therefore I should include it in the database, definitely. I think Sesame is probably going to have a pretty good annotation. I just believe this. And finally, we have Erythrantha gutata, the monkey flower. So with these 4 nearby neighbors, these are the closest annotated genomes we can find. We can grab down these protein sequences, and then when our transcript hits one of these in BLAST, we can feel pretty comfortable. We're going to try to use the same reciprocal best hit rule. We feel pretty comfortable about transferring that annotation to say, this is a putative, whatever you want to call it, glycolysis protein, whatever. So we have this option then of using existing annotation information to let us know whether these transcripts have likely functions through homology. Now, just because I'm doing this in Chia doesn't mean I'm wedded to doing this entirely in plants. And I have to say, the world is running out of fuzzy animals that haven't been sequenced. If you start looking through the outback or through the veldt, either way, you're going to run into lots of creatures that you think, well, that's probably not been sequenced, but it has. It really has. If you start looking at maybe the black rhino, no, the black rhino has been sequenced. Has it been? They haven't made the annotation public, but it exists. What about the Sumatran rhino? No, it's been sequenced. Elephants? Sequenced. Giraffes? Sequenced. Hyenas we can do. Hyenas we can do. There's a wildlife TB group in my division, very helpfully, and they've discovered that hyenas are very resistant to infectious disease. Think about what these things eat. How could they possibly be? I mean, I guess they would have to develop. There's a strong selection for them to have a great power to avoid infectious disease, considering what they eat. So we thought this would be a great creature. We take a look through UniProt, we get 162 proteins. It's got nothing. So what if we use the same pipeline that we developed for Chia and apply it here? Can we start with an RNA-seq data set, here produced at CPGR, on a much better instrument, the NextSeq 500? Can we build a de novo assembly of its transcriptome, and from that, start doing proteomics to back up this infectious disease research? I'll bet we can. I'm really excited about what comes out of it. I have an honor student on it. I have great faith in him. It's going to be exciting. Okay, so there are some takeaway messages. Proteogenomics is not one thing. Proteogenomics is a growing field, it's a confusing field. It means a lot of things to a lot of people. And I hope that walking through these three big thrusts, you can see some of the different ways that proteogenomics can help your research. Secondly, bioinformatics frequently requires side trips into biostatistics to offer its full value. I think that most of the departments I visit secretly suspect that they know less about statistics than they would really like. But the reality is we're all struggling together on this one. So I pointed you to my YouTube channel a moment ago. I hope that when you go there, you'll find a whole lot of lectures that help you in understanding how things like ANOVA work, or maybe even principal component analysis. These are valuable tools. And understanding how they work is very nearly a prerequisite to understanding what comes back from them. I really strongly suggest you bone up on all the biostatistics you can. Okay. Non-model organisms have been sort of a no-go zone for a lot of molecular biology. But that day is rapidly coming to an end. We're sequencing everything these days. Sequencing ribose somewhere, I'm sure they are. So you will probably find that if there's an organism that you find interesting, there's probably someone out there developing the molecular information needed to manipulate it and to understand it. So non-model organism, I think, is a term that's going to be progressively moving further and further out of date. So I have a lot of people to thank. First off, my division at Stellenbosch University, we're the folks out at Tygerberg campus, not the ones way out in Wineland. But the Division of Molecular Biology and Human Genetics has been a really good home for me for the 2 years that I've been here. I am very appreciative of my chance to work with the University of the Western Cape Department of Biotechnology. For heaven's sake, they allowed me to try out teaching statistics for the first time, and that's kind of scary for a guy who's really a bioinformaticist, so I thank them. The Center for Proteomic and Genomic Research has been a really helpful set of collaborators. They produce some pretty reliable service out there, and I'm really looking forward to seeing what they've produced in this next gen sequencing experiment. And finally, the South African Medical Research Council agreed to pay 60% of me so that I could wander around and talk bioinformatics and biostatistics with people. So thumbs up on them. That was a good risk to take. So, and finally, thank you for allowing me to come talk with you today. Thank you. They told me to keep it short in case somebody had questions, so I'll try to do that.
Thank you, David, for making bioinformatics interesting. Not an easy task. Does anyone have any questions?
When you talk about orthologs, about orthology analysis,
yes.
You said that it's reciprocal lasting.
A reciprocal best hits, yes.
Yeah.
Mm-hmm.
And then how would you resolve if, for example, the 5 top hits have the same p-value?
First off, e-values tend to be a bit of a mess when you have close relatives. And in the case of sorghum and maize, we found that we were frequently running into very low e-values, kind of suspiciously low e-values. So we focused on bit score instead, which is the number that gets translated into that. Back when BLAST was first created, it was initially published using bit scores to decide how closely related things were. And it was very good for ranking things, but people wanted to have a better way to say, how likely is it that this top match comes from random chance alone? And for that, we really needed to translate to e-values. In any case, we ended up using bit score thresholds of 50, I believe, for these two proteins to be called similar at all. And we only allowed for the best match. for each hit. So if this gene shows similarity to 5 different proteins in the other database, we only take the one with the best matching score.
But what if, for example, the difference between the first and the second bit score was like 1 unit?
In this case, that was enough to exclude from consideration the second hit.
Okay.
It's a very conservative rule. It could potentially lose hits as a result.
Yeah, you could increase a lot your final data set if you allow for
Sure.
some.
Actually, in the United States, some folks have argued that the way we do voting has kind of similar problems. That if no jokes about the current president, he's not going to help us. No. If people were to say there are these 4 candidates running, and I pick this candidate as my first choice, this candidate as my second choice, this one as my third, and this one as my fourth, you end up with a whole lot less penalty for not being associated with the majority party, for example. This is one of the forces that keeps the United States in 2 parties all the time. So we, in this case, I'm using a very conservative rule because I didn't want to claim any 2 proteins were similar when they weren't. But the cost of that was sensitivity. There were some linkages between proteins that I had to reject because of these very strict rules.
Yeah, and this way you also don't account for duplicates.
Right, yes, you may have paralogs existing for the gene in this species and only the one in the other. And this rule will force it to match to one or the other, but not both. Absolutely. Yeah, it's a very real concern. We've looked very slightly at what happens if we get more expansive about our association rules, but we haven't done it in a rigorous way yet. Thank you, good question. Yes, sir.
So as you mentioned, a lot of the plant annotations are rubbish for non-model plant species. So with your CHIA project, how are you going to avoid just transferring that rubbish to your IC data?
Yeah, that's a good question. Typically we use weasel words in bioinformatics, and one of these is putative. So when you look up a particular protein sorry, I've stepped on a toe, haven't I? If you look up a protein and you see that its function is putative such and such transporter, you should just keep in the back of your mind, oh, that was probably transferred at some point. And it may not be backed up by biochemistry. So it's better than having no information whatsoever. We also propose having a little bit of backup. I didn't really talk about it here, but the use of interpro. So interpro is not a whole sequence aligner. It's not like BLAST in that respect. Instead, it's looking for little motifs that are associated with particular functions. So if this section of 20 amino acids is known to be associated with leucine zippers, then knowing that's there is better than saying, I've got nothing on this sequence. I have no idea what it does. So some of the hits that you get back from interpro are really uninformative. Like something like a third of our database matched to disordered region. That doesn't help me very much. But if something says similar to RIP domain, I'm going to say I know something about that region, right? This is likely a ricin analog. So yeah, you get different amounts of information back from these hits, but we're trying to go both the whole sequence route and the motif route to give us as much information as possible.
And are you going to weight the BLAST annotation based on whether you get the same kind of annotation from 1, 2, 3, or 4 of your relatives?
Oh Lordy, that is a tough thing to do. I don't think that's going to be so feasible, mostly because of our very restrictive reciprocal best hit rules. So we're automatically throwing out anything that isn't the best match. So we have these 4 reference species. If our best hit is to the carnivorous plant, then I'm not really going to pay attention to what the second, third, and fourth best hits would be. If I have a student who can take on this project, they'll probably spend some time looking at that before saying, I'm not sure what to do with this. I'm not sure what to do with that, in short. And one of the things that a proper sequence biologist, a sequence bioinformaticist suggested to me was when doing my maize to sorghum comparison, throw in an obvious ringer, a species that's further out and outgroup. So we threw in rice as a third alternative. And sure enough, we got fewer hits between rice and sorghum than we did between sorghum and maize. So their evolutionary their presumed evolution is quite useful in predicting where the best hits are going to come from. Again, good questions. Yes?
Your proteomic data versus your genomic data. So the genome doesn't change depending on the conditions that you're growing your bacteria in. The proteomes that you've got from your different teas, what conditions were they grown in, and how likely is that to be similar to a real lab situation?
These are mostly just exponential growth. I mean, these are not, we're not putting them in oxygen-starved conditions, you know, nothing to emulate what it would be like if they were going into a more dormant state in somebody's lungs. So these are just trying to go just as deeply as we can. How can we rapidly get as much protein mass as possible? So we were not really investigating to a great extent the quantities of how do we see particular proteins that were regulated up quite a lot in this strain, but regulated at a lower level in the others. We didn't do anything of that sort because the information wasn't really that valuable in this case. We didn't have these same bacteria growing under different conditions to do that kind of analysis.
Yeah, I guess what I'm asking though, if you're looking for those transcripts, if you're trying to identify which part of the genome is actually coding, are you growing them in enough conditions that you're going to get those rare things expressed?
No, we were going to get the ones that are expressed when everything is on. I think in this case we saw approximately 2,000 proteins, which is not the full 4,400 that we could get, but the other question was, is the mass spectrometry we're doing here capable of the same sensitivity that we could get if we were doing fractionation in labs that use it routinely? And I think the answer there was no. We actually had a pretty big sensitivity hit compared to what some of the labs that have, for example, Rudy Aebersold's group at the ISB has run 24 fraction sets on a very similar kind of instrumentation. And their sequence depth in terms of number of proteins wasn't hugely greater, but the number of peptides they saw per protein certainly went up. So this is the quantitative measurements that we were able to make were somewhat restricted by the nature of the way the experiment was set up to begin with. These experiments were not captured to begin with the assumption that they would go the proteogenomic route. The data were all collected in 2012, and our publication is last year. So we were trying to find a way to turn this large data set we produced into something that actually had a really compelling story to tell. And by combining with the genomic data, we were able to find a way to tell what we think is a pretty compelling story. And we passed peer review, I'm happy. So, yeah, good question.
Are there any other questions for David? So I'll just ask one last one. So how often do you see proteins that don't hit back against the genome or protein? So you spoke about the opposite.
That's a very interesting question. So how frequently do we have spectra that we fail to identify? And the answer is every experiment. Every experiment, a huge swath of it. This is what a lot of people in my field have taken to calling dark matter. So that one of the questions you might ask of anyone doing a proteomics experiment is, what fraction of your tandem mass spectra are confidently identified to a peptide sequence? And if they answer all of them, they clearly don't know what they're doing. Because the answer is usually, best case, about 50%. So why is that? Why is that? So there are lots of arguments for this. There are lots of people who will tell you there's lots of gold there and we need to just dig into that and figure out what it is. But in fact, what we see is that our instruments are so good at producing tandem mass spectra, just pouncing on any peptide that's showing up, that frequently they're digging into a very, very sparse signal to try to produce those fragment ions. And you have so little signal caught in the trap for producing this tandem mass spectrum that you simply don't get a very good signal-to-noise ratio. And if it's all just static, your matching is going to match it to whatever it can, but it's all going to be with a very low score. The other possibility is that there's gold in those hills. So it could be that we are not using the post-translational modifications in our searches that correspond with that particular peptide. We could lose it that way. We could lose it because we didn't have the right peptide sequence in our database. So if, for example, I'm working with sputum from someone's lungs, and I include MTB and I include human in my database, any other respiratory infection they might have would produce a peptide that I can't identify because it's not included in my database. So there are contrasting ideas about it. I would say that our field has gotten a whole lot better about identifying spectra when we collect them, both because we have controlled when we collect tandem mass spectra to those isotope packets that look like they're going to be identifiable, and by producing high-resolution tandem mass spectra where we have fragments that are accurate within, say, 20 ppm. When you have that, it's a whole lot easier to identify those. Our identification rates go way up. But it used to be in the bad old days of just ion trap everything that we could identify 10% of the spectra in our experiment and count ourselves lucky. So the field has gotten a lot better in this, but we're still producing far more spectra than we identify in every experiment.
Any last questions? Thank you very much, David.